Good morning. As she said, I'm Kyle Pinheiro. I'm a PhD student at Duke University. Today I'll be discussing some work that my lab, the lab of Dr. Luca Gordon, has been doing related to cooperativity between transcription factors with neighboring DNA binding sites. I'll discuss some experimental methodology we developed to determine cooperativity, as well as some work we've done on computational modeling. Throughout the eukaryote genome, we see regions where there are pairs of neighboring transcription factor binding sites. We know from previous studies that in some cases, the occupancy of one of these transcription factor binding sites affects the binding affinity of the other transcription factor to its binding site. It is not, however, implicit just that because there are two neighboring binding sites that they will in fact bind cooperatively. And so we set out to answer the question, for any given sequence context, do neighboring transcription factors influence one another's binding? For the purpose of our study, we focused on the ETS1 transcription factor. Throughout this talk, I'll discuss primarily our work with ETS1 RUNX1. We did also do some work with ETS1, which I will mention very briefly a little bit later, but for simplicity, ETS-RUNX will be the focus. We used ChIP-seq to identify regions throughout the genome where there were neighboring ETS and RUNX binding sites. We then synthesized these regions onto a protein binding microarray. And as with the standard protocol for a protein binding microarray, the way that this works is we're able to measure the ETS1 binding level because it correlates very closely with the fluorescence that is associated with an antibody tag that we have that is attached to the ETS1. We went about this by creating two different scenarios for measuring ETS1 binding level. We measured the ETS1 binding level in the presence of RUNX and in the absence of RUNX. And simply put, once we had made those two measurements, if the ETS1 binding level with RUNX was with statistical significance greater than the ETS1 binding level without RUNX, we judged that, we classified that as cooperative. If there was no statistically significant change, we discerned that to be independently bound. Here, I simply show kind of a summary of the data. In green, we have the cooperative, those points that were deemed to be cooperative, and in blue, those deemed to be independent. We had around 2,200 data points that were each the result of several replicate sequences on the array. And it was roughly half and half cooperative and independent. We wanted to model the cooperativity, though. We wanted to be able to, given a small sequence with two transcription factor binding sites, discern computationally whether cooperativity would be exhibited or not. First to do this, we designed several features that were, from previous studies, found to be biologically important to predict cooperativity. Distance between the binding sites was one, the orientation of those binding sites, and also the binding strength of the transcription factors to the individual binding sites. We additionally looked at DNA shape, and of course, we could use a raw sequence or one hot encoded sequence. The classification targets are very simple. The cooperative, we judge each sequence to likely exhibit a cooperative interaction or independent binding. We additionally wanted to look at regression, and so we defined a regression target as the delta, the change in ETS binding level between the with RUNX condition from the no RUNX condition. For the classification task, we started with random forest regression, random forest classification. And here I have a chart of the AUCs. And the particular trend I want to point out here is that this is several different combinations of features that we used as input for several different models that we trained and evaluated. And the key thing to note is that the strength feature was the most valuable. We're able to get up to a 0.88 AUC with the strength, and no other combination was able to exceed that by very much at all. This is the one place I'll also reference our ETS work, because I think that the contrast helps to show some of the trend here. Here is some of the curves for the ETS1 models. Unlike the ETS1 RUNX1, the ETS1 RUNX1 models, we see much higher importance on distance and orientation, where strength alone doesn't attain nearly the highest AUC, whereas distance and orientation together attain almost the highest AUC. And so between these two, we get a sense of the characteristics of the cooperativity mechanism. And these can be mapped back onto things we know about the mechanisms from previous studies. Simply put, ETS1 RUNX1, because of its mechanism of cooperativity, has some flexible characteristics related to regarding distance and orientation, whereas for ETS, those characteristics of distance and orientation are more rigid. It's more important that they are in a certain position adjacent to one another. As we moved to regression, we first attempted to use random forest regression for that purpose. We also worked with support vector regression. And in neither case were we able to attain the results we would hope for in terms of R-squared values. We shifted our focus towards convolutional neural networks for that reason. We felt additional representational capacity could raise the ceiling on the accuracies we're able to attain. Our model loosely was based on DeepBind, which came out of DREAM a number of years ago, and many of its successors operated by essentially just adding deeper convolutional networks. And that is essentially what we did. We did a variety of grid searching over the ideal architecture, and what we end up with is a one-hot encoding of the sequence, several convolutional layers, fully connected layer, and output our prediction for the change in ETS1 binding level, which is our proxy for cooperativity. I'll note that in addition to the sequence feature, which is the most important in the neural network model, we can also add in some of the derived features if we so choose as input to the fully connected layer. We did, as I said, we did a variety of grid searches to identify the best architecture. One aspect of that was ensuring that we had the proper depth of the convolutional layers to pick up the somewhat complicated patterns that we anticipated there being when we were dealing with multiple transcription factor binding sites and the relationship between those. And we see a trend where as we increase the depth of the convolutional network, we see improved R-squared with some mild drop off moving from 4 or 5 to 6 and 7. However, we thought we could still do better because this gets about 0.57 R-squared. And so we added in, as I had described, we added in binding strength as another feature to the fully connected network. And this across the board, regardless of how deep the convolutional portion of the network was, we attained a sizable bump, which is interesting because we would hope that based on the sequence alone, it would be able to pick up the information that we could give it via binding strength directly. But it seems that it's not. There's still more information that it can gain just from being directly given the binding strength for the binding sites. To summarize a little bit of what we just looked at, here we have a chart where in blue, I have random forest regression, our squared results, and in green, convolutional neural network. As I suggested on the past slide, just between the two convolutional neural networks, we see a great benefit when we add binding strength to sequence. That's also what we saw when we were looking at classification results. And if you take a look at random forest regression, we see the same thing. Strength is very predictive in this case, while distance and orientation less so. Additionally, it's interesting just to say, with random forest regression, sequence alone did not do very well, whereas convolutional neural network with sequence alone did fairly well. So we're able to extract a lot of information directly from the sequence when we use a convolutional network, as we would hope. And as I described, we see a lot of benefit from using strength, which, as I said, maps well back onto our understanding of cooperativity of ETS-RUNCs from earlier studies. However, the biological insight of the cooperativity mechanism is not really what we're delivering here. We've presented a method for quantifying cooperativity between neighboring pairs of transcription factors via our protein binding microarray assay. And we've shown that we can train accurate models, both for classification and regression, such that those models reflect biological insights that match lower throughput studies that we've seen, and that we can predict cooperativity for sequence contexts that we have not actually measured in the lab. The future work then is to apply these methods to other transcription factor pairs, just expanding our knowledge of transcription factor cooperativity and its various mechanisms, and also to experiment further with the modeling. We don't believe that we have hit the ceiling of what the models can attain in terms of our squared on these data sets. Further work could go into adding some recurrent neural network components, as well as some interpretability components. There is a paper that should be on bioRxiv in 2 or 3 weeks. Maybe going back to my hotel room to work on that after this. That is first author Vincentius Martin, along with several of my other lab mates, whom I would like to thank. They contributed to this project or listened patiently as I described it and gave feedback, along with my project committee and advisor. So thank you. I'll take any questions. I can hear you. I don't know if I can hear the mic.
Okay, so it was exciting to see you trying to assess the effect of distance on the cooperativity, and people in the past have tried to test this sort of models on much more minimal data, and have not been able. One of the things people wanted to see if there are nonlinear dependencies on distance, periodicity and so on, but mostly none of those approaches have been successful. So I was excited that you will find, you have the data set to actually test if there are interesting nonlinearities in the distance dependence. Is that something that is captured explicitly in the CNN somehow?
I would say that is something that we can't quite tell the way the convolutional network is set up. That is one of the reasons why a next step would be to look at ways to make the whole thing more interpretable. So yeah, in answer, we can't quite tell based on the way the convolutional neural network is set up.
Okay.
I will say not on the basis of the model, but on the basis of our data. On the left this is all data purely from the experimentation, not from the model. And looking at the bottom left, knowing what we know, you can kind of read some periodicity into it. that's not a super objective thing. But the interesting thing, at least in this case, kind of going back to what we know about the mechanisms for ETS-RUNCs and ETS-ETS, is that given the flexibility of the ETS-RUNCs interaction, it makes sense that there's kind of a level, there's kind of a wide range within the range we tested where you see cooperativity, whereas in ETS-ETS, you do see a kind of a drop-off that reflects the rigidity there.
Great. So there is a remote question. Because this is the first one, I would like to prioritize that. So the question is from Ignacio Ibarra del Rio. And the question is, great talk. Could ETS1 binding as dimers in your oligos still occur? And encoding of those might potentially boost your final performance. There's a second part to that. Do you want to?
I'm sorry, I'll need the question to be repeated.
Okay. Could ETS1 binding as dimers in your oligos still occur? And encoding of those might potentially boost your final performance.
Could I see the question? Okay. Still encoding, those might potentially boost the final. Perhaps. I think not. I think that one of the things I didn't describe is kind of the normalization we did for the sequences that we used. We did include some negative control. And so if there had been other ETS1 binding sites that we kind of overlooked, that would have come up in the negative control and we would have taken that into account. And the second part of the question is, do we compare against related regression-only models, e.g. feature reduce and or probound? I'm not familiar with those two models.
So I was really surprised by your result that adding in the strength to the CNN so much boosted performance. That's not been my very limited experience. I was wondering if you looked at the specific exactly where the model's improving. Like, do you see that in general, your model's getting a little more accurate across most of your examples? Or are there a subset of examples where your prediction's just gotten way more accurate? And if it is a subset, what are the examples where the strength is really helping?
That's a good question. We did not investigate that specifically.
Yeah, thank you for the great talk. So, so this assay basically is used for measuring the cooperation between the transcription factors, but I'm thinking about the other way. So for like these adjacent transcription factor binding sites, can they actually negatively impact each other? Like because they're too close and one binding of one transcription factors might affect the binding of the other transcription factors. So, because as you show in the previous slide, like with both transcription factor binding sites present there, and if you minus the strength of these with the both presence there, and you get like probably like a negative strength, positive strength as compared to one binding site, but can it be negative as well?
So the question is then, could you say it a slightly different way? Okay.
Can there be negative cooperativity?
Oh, yeah, competition?
Yes.
Certainly. We just adjusted, we adjusted our sequences such that was not a huge issue. This data, you can, in this data, you can see there are a few things below the line. That's likely noise. Technically, that would refer to competition. And so while the dots below the line can be interpreted as competition, or it could be interpreted as noise, it could also be interpreted as competition. Our lab does other work specifically related to competition. But so, yes.
Thank you.